So let's welcome our Everson lecturer, Art Edison.
Thank you, Chad. I really appreciate the overly generous introduction. In fact, just to set the record straight, I am not the CCRC director, but I direct the NMR facility within the CCRC. So the most important part of the CCRC. Yeah, so I am so honored to be here in this capacity. You know, I was here for 7 years. I love Madison. I love this campus. Lived in Eagle Heights, U houses. And I have such fond memories of this place, and it really did me well. So I'm very happy and proud to be back here. I also want to just set the context. I know that many of you, if you do metabolomics, you're probably more familiar with mass spectrometry as a method for metabolomics. And I realized I even didn't put in a slide contrasting the strengths and weaknesses. Mass spec is much more sensitive than we're going to be talking about, but NMR is much more quantitative and has a huge amount of advantages that I'm going to try to highlight in this talk. And I will show you what we're doing with NMR that I think is useful. So I'll start with defining the problem. And we have the other thing I want to say from the start is that I'm not going to be highlighting a lot of the biology. We are interested in biology, but we've actually recently come up with the technique that I'm going to highlight in this first half, and I'm really going to talk about the technique, especially with that. But we work on Neurospora crassa. This is a filamentous fungus that actually Beadle and Tatum used for their one gene, one enzyme hypothesis Nobel Prize winning work. We love Neurospora crassa. We have a lot of interest in Neurospora crassa, and right now I'm just using it for its spectrum and for its complex mixture of metabolites. But I'm gonna keep on the Neurospora crassa theme for just a little while. So this is the kind of thing that we measure in NMR metabolomics. And any of you who have done any of this sort of work can recognize this. It's basically a collection of a bunch of organic chemicals all mixed together, and we're trying to figure out what's what. And so just to highlight a little bit here, I'm just going to expand this region and show you that, you know, so there's some big prominent peaks somewhere. There we go. Some big prominent peaks, but then there are also a lot of little teeny peaks down here that are actually real. And sometimes, you know, I say that they're real because I know that they are from work that we're going to get to in a little bit. But some of these you wouldn't be confident to interpret directly from the spectrum. They look like they could be noise or artifacts, but I'm here to tell you that they're not. And there are a lot of mixtures in here. So the problem that we face in NMR metabolomics is that we have to figure out what is in here. We do a lot of 2D NMR, which helps quite a bit in annotation and identification, but a lot of people work directly from 1D proton NMR spectra. And I say that I think it's a little bit risky if you do that without some additional measures. So for example, I'm just going to highlight some compounds that I know because of what I'm going to show you later belong in here. So here's valine. Here is isoleucine. You wouldn't necessarily know it, especially in some of these resonances in here. You can't find those in the mix. GABA, ornithine, alpha ketoglutarate, malate. Some unknown that we know is a molecule, but it's in here. So it is really difficult looking at this spectrum of the mixture and trying to decide what's inside that mixture. There are different ways that people have proposed to do that. And without being too bold, I think that we figured out the way at least that I'm most comfortable doing it, that I'm going to tell you about next. So overlap makes quantitative NMR extremely challenging in metabolomics. And this is one of the moments to introduce this network for advanced NMR. And I'm sure that many of you here know about it because we're partnered with University of Wisconsin and the University of Connecticut. And it was very satisfying for me because it was sort of full circle. I did my PhD in NMR FAM back when it was not in NMR FAM as you know it right now, but it was a different building. But close to probably right here. And so and John Markley was involved with the initial discussions. In fact, he came up with the acronym NAN. And so that we owe that to him right now. And so but Chad and Katie have a 1.1 here that I'm sure that you've all seen, and it's dedicated largely to solid state NMR biosolids and materials. We have a sort of the sister 1.1 in Georgia. It's a very low resolution picture, sorry about that. And ours is dedicated to solution NMR, metabolomics, and structural biology. We have all sorts of other infrastructure. The biggest one is that all of the data from every instrument in our facility gets transmitted directly to UConn and stored in a server and then is made publicly available. So data management is really a lot easier because of NAN. And then, and now I want to just take that opportunity to talk about the resolution that we're getting. So this is, so the 1.1 gigahertz of take-home message that I want to highlight in terms of the 1.1, at least from the solution side. There are different strengths and weaknesses on the solid side, but on the solution side, the proton sensitivity is nothing great, but it's not bad, but it's not the thing that we're most pleased with. The resolution, the spectral resolution, is really what we are extremely excited about. This is a spectrum of glucose at 600 megahertz, and this is the same the same molecule at 1.1 gigahertz. And you can see we get much better spectral resolution if you just look in some of these regions right here. But more important, the strong coupling effects that you get by having chemical shifts very close together and about the same size as the coupling constants, those are reduced at 1.1 gigahertz relative to 600 megahertz. And so this is just the easiest example to demonstrate, and I'll come back to some other glucose spectra in a few minutes, but basically, so one of the real strengths of this kind of resolution is for carbohydrate analysis, and every carbohydrate basically looks like this, the 1.1 will really help that field quite a lot. So it improves spectral resolution. But we still have overlap problem for metabolomics because we don't just have glucose in our samples. We have a bunch of other stuff. And so that's where I'm going to talk about first. And I'm going to tell you about what we're doing in this topic called metabolite fraction libraries. This is all unpublished, but within the next 1 to 2 weeks, we'll have a bioarchive up on this. It's almost complete. So Chris Esselman is the graduate student, bioinformatics graduate student, who's done all of the work in this area. It really has been a phenomenal job. But to set the stage, I'm giving you this little snippet of a spectral region of NMR spectra. And I'm highlighting, this is all just a mock-up, but highlighting two different groups, group 1 and group 2, that you might want to compare one of them is disease, the other one is normal, let's say. And there are different ways of tackling this problem. For many years, so here's the stage, and we want to know what's the difference between the red group and the blue group. For many years, our lab has used a chemometric approach. And in fact, until just recently, this is basically the approach that we would take. Chemometric essentially uses statistical methods. Before we start to analyze the spectrum and figure out what's what, we'd use statistical methods that will then tell us through a PCA scores plot or more complicated multivariate analysis and various loadings plots or other kinds of analysis, we can tell what compounds are associated with what groups. And so that comes out pretty nicely with chemometric analysis. There are tons of tools available. This is the simplest PCA scores plots and loadings plots, but there are a lot of sophisticated tools that have been developed to try to figure out the important peaks. So the key here is that you are figuring out what's important to your study before you go through the analysis to figure out what the peaks are. So that's the big advantage in my mind in chemometric analysis. But now I'm going to highlight the peak over here, or this region over here in black, and I'm going to introduce the concept of quantitative approach to metabolomics. And so And there is software out there that tries to do this. And some of you might be familiar with Genomics, a very popular commercial software package that will fit libraries of compounds into NMR data. This is kind of that same concept. So here we have the original, the actual spectrum is shown in black for one of the samples that I highlighted on the last slide. And then these are compounds that Chris put in a simulation that make up this sample. So we know what they are in this case. And this black is made from the summation of all of these different compounds, ornithine, lysine, arginine, and GABA in this case. So you can see the amount of overlap in here is pretty tremendous. And you might, you know, in a software tool where you just have a library, maybe the library says, yeah, GABA is the best fit right here, but might be a little bit iffy. You might not be sure that that's exactly the right thing if you're not careful. So it's a little bit treacherous with to just look at that mixture and decide that you know what's there. So this is the problem that I'm going to try to attack in the rest of this section. Okay, so we are introducing this concept called quantitative NMR metabolomics without databases. So this is the key thing. So if you do a genomics analysis, one of the things that you're buying is a large database that they have available, and it will try to find the best fit, and you visually look at it and determine if it's the best fit. You can adjust things back and forth, but you have this large database to work from. People tend to always think the bigger the better when it comes to databases, and that's the same with mass spec. If you get a mass spec signature, you have a database with 1 million compounds in it. Your chances that your compound of interest is in there increases, except so does the false discovery rate. So the bigger the database that you're searching for, the more often you're going to be finding inappropriate matches. So that's really a problem with databases. If they're big enough to hold all of the compounds, they might be too big to give you confidence in the matching. So this is what the approach that we are taking instead. I'm going to show you, this is the overview of the whole thing. So we have, our first step is that we take a mixture like the Neurospora crassa and we do old-fashioned HPLC. So another, a little, sort of, I guess, word that in this one is that basically I think that NMR has been, we like to brag about the fact that we don't need sample preparation, and that mass spec has to do so much sample prep upstream because it'll clog up the LC part of the analysis. Well, I think that the reason mass spec is so popular is not just because it's sensitive, but because they separate their molecules properly. So, and NMR has been avoiding that because we don't need to do it. We can get nice spectra mixtures, but then we just don't know very much about them. So if you don't learn anything else about my philosophy right now, it's that I think that separation is better at the start, and I'll show you how we're approaching that. So we separate our mixtures. We just do this once for a study. This whole process right here takes about a week. So it doesn't, if you're doing a big study, it's not that big of an additional bunch of work. So we separate our mixtures with HPLC, we collect fractions, and then I will tell you in a few minutes about this technique that we use to process the data that we developed with Frank Delaglio called SAND. It's time domain analysis. And then, so each one of these peaks is actually separate Lorentzian peak in there. So we got them separated in both chemical space and frequency space. The next step is that we correlate all of these peaks across the eluting fractions. And I'll show you all of these steps in more detail. So wherever a peak first shows up to where it ends, we take that and correlate it across those fractions. And we create what we're calling a metabolite basis set. This basis set contains every single correlated set of peaks that are in that sample. So if you will, we're like recoupling the metabolites. We've pulled them apart with the fraction library, and then we recorrelate them into molecules. And the correlations are very, very high. So we don't have to mess around with thresholds. It's almost perfect correlations. And we get this group of molecules that we're calling the basis set. We don't know for sure that these are all legitimate metabolites, but we think they are. And we think that this forms the complete set of observable NMR metabolites, whether they're known or not known. That's one of the key important factors in all of this. And then Chris wrote a package called FT Analyzer that does that work, or FL Analyzer to do that work. And then we can match databases with these, like the BMRB. We use that a lot. Step 3, we use a package called Batman, which is a Bayesian tool that Tim Ebel's group at Imperial College developed to fit our basis sets, that's our database at this point, into an experimental spectrum. And so we take an experimental measurement, let's say in this black right here, and then we fit our basis sets into there to get quantitative amounts in the profiled unfractionated experimental spectrum. So that's the overview of the technique that I'm going to walk you through right now. Feel free to interrupt with questions along the way if you want, if that makes sense. Okay. So here is the same N-Crassa extract that I showed you before. On the top is the unfractionated mixture, part of that I also showed you at the start, and then the y-axis are 140 separate fractions that we collect in our HPLC. And then we actually have a single quad low resolution mass detector that we can collect a chromatogram, because we're going to do multiple injections, and we want to make sure that these injections, that the chromatography doesn't change from injection to injection. And then here's what you get. And I'll highlight a few different regions. So if you will look at, for example, the peak right here in box 1, nice sharp set of multiplets, totally obliterated by whatever is up here. You would never see that in a 1D spectrum. You might see a little hint that there's something that's not quite, you're not sure what it is, but you would not see that. But this is a beautiful separated, nice looking molecule, whatever it is. Box 2, this pattern and this pattern right here, they look the same. So they look like those two things are probably in the same molecule. They appear to be correlated across the fractions. But if you also look up at this part of it, it's obliterated by everything that's up here. You wouldn't see that at all. Over here, it's overlapped with a different set of molecules. You wouldn't see that. Point 3 is actually something that I find really intriguing up here. Like if I looked at that peak in a 1D spectrum, I'd say, oh, the shimming's bad, or the phase is off, or something. I'd complain to my student, I'd look at that and say, yeah, it's kind of weird. But what you see instead is that you have this pattern of overlap that accounts for all of that weird looking peak shape that you see in the 1D spectrum. And then the fourth box down here at the bottom is basically highlighting a bunch of tiny little peaks. And if you look up here, you can kind of squint your eyes and maybe see that those peaks are up there, maybe not. I'll tell you why they probably actually aren't up in the upper one, but they are in the bottom one. But we can see some very small peaks down in this fraction library, and those are real, and they can be correlated, and they've made into basis functions as well. Okay. Now, sensitivity-wise, and this is related to these small peaks, to quantify the sensitivity. So first of all, let me say that we injected about 15 times the mass of Neurospora into our HPLC, as I'm using as the example trace to show for the unfractionated material. So 15-fold excess, because we just do multiple injections on the HPLC and then collect the same fractions in the same tube. So the peaks right here in green are histidine, one of the peaks that's free that we can integrate in the unfractionated spectrum and also see clearly in the fraction library. So it elutes from fractions 110 to 117. You can see it goes up, it goes down. Actually, in the middle, it has a dip. Sometimes we get that in the chromatography, and if anything, we're not sure why we sometimes get these dips, but we do, and if it even makes the correlations even better. So we sort of embrace the dips. And then in the back is the mixture right here. So all of these have DSS in the spectrum at 3 millimolar concentration of protons. And Chris integrated the values of each of these peaks compared to DSS. And what you find is that, so in this fraction 110, it's 33 micromolar. In fraction 117, it's 27 micromolar, pretty low concentration. And these are about 50 microliter tubes, so 1.7 millimeter tubes. So there are just a couple of nanomoles of sample in here. So the thing that we always complain about, that NMR is so insensitive, is not so much so true. I mean, if you do it right, I mean, basically, you know, this is between 1 and 2 nanomoles of sample right there in that spectrum. The mix is 170 micromolar. And if you sum all of these peaks together, you get 2594. And actually, it's embarrassingly close to 15. I think it's 15.2 times the mix. So it scales proportionally to how much material you injected onto that experiment. So if you want to increase signal to noise in this tractor technology sort of approach to metabolomics, you just inject more sample in the auto sampler, in the HPLC. Okay, so then I have to highlight the fact that we also save about 10% of every fraction for mass spec or for functional testing. So then we can start to associate every fraction with other technologies or functional information. I'm not going to show you more about that today, but that's a very valuable part of it. So those of you who know NMR know that 2D NMR can do some of this stuff. You can correlate peaks with 2D NMR just fine. That's kind of what it's invented for primarily, is to find these associations. But you can't, for example, just test a fraction with 2D NMR. And then I think that these fractions have a lot of value to them in their own right. So what do we do with them? Well, I'm first going to take a little diversion into sand. This is the work that we did with Yue Wu in my lab and Frank Delaglio and a few other folks. But basically we took, and Frank is really the intellectual driver on this project, but you break up so NMR is collected in the time domain, and then it's normally Fourier transformed into the frequency domain. And one of the problems with the Fourier transform, as wonderful as the Fourier transform is for NMR, one of the problems is that if you have points that are points that are distorted early in the FID, it leads to baseline distortions in the Fourier transform. All sorts of distortions in the FID can cause all kinds of baseline problems in the FT. That's why I came to Frank and I said, Frank, I'm sick of these baseline problems that we're having in our proton metabolomics spectrum. He said, well, I've always been thinking about this time domain method. Why don't we try that? And so the problem is that we don't have computers that are big enough right now to just directly analyze an FID and get all of the frequencies out of it. So what we had to compromise on, we basically do a pilot Fourier transform, cut a little chunk out that has 3 to 5 peaks in it, take that chunk to the inverse Fourier transform, and then model that time domain data with an exponentially decaying function to give you a pure Lorentzian peaks. You fit the pure Lorentzian peaks, and then you can determine what the RMS deviation of this is over how many peaks you add into the signal. And when the RS deviation doesn't improve, you've added enough. And then you end up converting you don't have the time domain or frequency domain. You now have something that we call the tabular domain. Actually, Krishnamurthy, who invented something called craft, is very similar. He was a co-author on this. He invented the term tabular domain data. So that we love the tabular domain right now, because we basically have number of points are the number of peaks that are in the spectrum. We have the frequency, the decay, and the amplitude. We would have the phase if we didn't just set the phase to 0, but those are the parameters that we get out of a sand analysis. And so then we get this peak over here in the frequency domain, ends up looking like that in the tabular domain. So not only do we have the table of peaks, but we have all of these peaks are separated into different individual Lorentzians. It works well. I'll point out the places that it doesn't work. These are some ground truth data sets that UA made, simulated data, where you can just focus on one of these panels, but basically the black is a mixture of all of these things up above, or one of those mixtures. The red is the sand fit. So the black is the Fourier transform traditional data. The red is the sand fit. The green are true peaks that UA put into the simulation. The blue are the sand modeled peaks that were obtained from that simulation. You can see a couple of places where it doesn't do it quite right. Like here, for example, down here, there's another one where sand finds an extra peak that didn't belong there. So sometimes you will find that kind of problem, but not very often. And it's oftentimes better than I would see as an experienced operator. The chemical shift correlations are almost perfect. What goes in comes out chemical shift wise. The amplitudes are really pretty good. They're better than this kind of plot shows, because this shows everything overlap regions. It's less good in overlap regions, but it's nearly perfect for non-overlap regions. And then the line width recovery is really good too. So we love sand. And then this is now a region of one of the fractions, fraction 110 from Chris's fraction library that I just showed you. And the black spectrum on the back is the experimental Fourier transform. The red is the sand fit to that Fourier transform. And then shown here are the individual Lorentzian peaks that contribute to that fit. So the key point here is that every one of the spectra in the fraction library are deconvoluted in advance with sand. So we have everything is an individual Lorentzian. Okay. So what do we do with it? So essentially just a brute force approach of correlating every single peak across every single fraction. And so I'm showing here on the left is a little region of the fraction library where Chris has highlighted, his software has highlighted 3 different peaks that are eluting over fractions. Actually, there are a couple of different peaks mixed in with these, but 3 different peaks that are eluting over fractions. So he finds the highest peak that's left in the fraction library, goes to that, and then he finds where that peak starts and ends, and then finds all the correlations, and then he moves down from there. And so you can just see here, those things look quite correlated together. They are well correlated together up here, quantitatively, essentially perfect correlations. If you're used to, Jeremy Nicholson published something called STOCSY, Statistical Total Correlation Spectroscopy. This is essentially STOCSY, but across the fractions. The problem is that STOCSY deals, you usually do it in unfractionated mixtures, and it requires, usually it's like 0.8 is a correlation coefficient that you're pretty happy with, or 0.9, maybe if it's a really good day, never this high. So because of the HPLC, because of the sand process peaks, these are essentially perfect correlations. When we get a group of molecules that correlate, we call that metabolite basis set N, whatever the N is indexed to at that point, put it into this metabolite basis set. Then we run a match against, we actually like to use, well, we love Gizmo, something that Milo invented with Hamid and Hassam here in this building. Gizmo will adjust the NMR spectrum according to the field strength. And so, but we also use it in its implementation that Raphael Buschweiler put Gizmo into Colmar 1D query. So you can query a Colmar 1D database set to the right. This is 800 megahertz data, by the way. And then you see what matches the best. In this case, glutamine is a perfect match to those peaks. And then, this is really this is showing one of the superpowers of sand, sand, if you will. So we take this fraction library, we find these peaks, we say, yep, that's pretty good. Then we make a second version of this fraction library with those peaks taken out of the tabular domain. So they're automatically checked off in the tabular domain. We reconstruct the spectra without those signals, and now we're starting off from scratch, and we go to the next set of peaks. And so this is nice because if you've ever had an NMR spectrum in metabolomics where you've tried to remove the water region, remove the ethanol region, something like this, you get all sorts of baseline distortions. Here, you don't. It's a perfect reconstruction. You just don't include it. So so that makes this really tractable and a pretty powerful way of doing it. So then we get to the point where we have this basis set. Now this looks like the fraction library, but it's not. It is actually ordered from the highest peak at the top or to the lowest of the point at the bottom. These are reconstructed metabolite basis set elements. And I'm just showing for ease a few of the database matches. So valine's up here, leucine, pantothenate. In the middle, we have an unknown, we get a lot of unknowns. And so the ones where we have a high degree of confidence in the match are coded in black. The ones where we have less degree of confidence, it's gray. Things that we really don't have any idea about are red. I think that was right. The gray and the red are, those two might be reversed, but I'm pretty sure that was the right order. So basically, this is one of the critical things about the method that I'm introducing here is that we have both knowns and unknowns in quantified metabolites. We're claiming that these unknowns are metabolites, but we have to still prove that. Okay, but we don't know what they are, but we can still do something with them. So this now is, I'm going to describe this fitting step, and I'll first show you the data with a so-called ground truth sample that we made. A student took 53 synthetic metabolites and made 10 different mixtures with different amounts of these metabolites. And so we have 10 profiling spectra of these things. This is just one of them. And then we made the fraction library with this mix and did all the steps that I just showed you. And so we created a metabolite basis set. So we only found 50 of the 53. So that was a little bit of a disappointment, but so for whatever reason, we did not find pyruvate or cysteine. I got to dig into that just a little bit more. This is like a week or two old result, and then we also missed galactose. But it turned out that we discovered after the fact that the galactose standard was degraded. She used a degraded sample in making these mixtures, and we characterized perfectly the degradation products or the contaminants in there. So those were actually, I consider that a win. So we'll consider this 51 out of 53. So down here in very low resolution are correlation plots. Essentially, most of these, with the exception of histidine. So this is like correlation of 0.58. Histidine did not fit very well to these, but the rest of these have 10 points that are correlated. So we took these metabolites, which I apologize for the resolution on this thing. But they're 10 or so metabolites that we were able to integrate directly from the mixtures, and then we take those integrated values and compare them to the values that Sand made or that Batman made in the reconstruction. So the red up here is the reconstructed metabolite basis set fit to the profile data. So we're taking those 51 with galactose's impurities, 51 basis functions, and fitting it to that spectrum up there. And then we have the residual down here with an RMS deviation of pretty small number. So it's a pretty good fit. And the ones that everything is, sort of 0.98, 0.99 sort of correlation coefficients, except for the more pH sensitive groups like histidine. And we think that we just have to adjust the parameters of the pH movement in the chemical shift a little bit better to improve that. Oh, now you can see it. Sorry. I should have done that one click. Here are the molecules, but you can see the fits more clearly right now. So I'm showing on the vertical axis the direct integration of the mixture spectrum, and then on the horizontal axis, the Sand-derived concentration. So we're fitting, or the Batman-derived concentration. So we're fitting that also to DSS in every sample as well to get a quantitative measure. So these numbers here are in millimolar. So it's a quantitative fit to the data. Okay, so most metabolites fit well. Histidine had a large pH range and had lower quality fit. And I'm just going to end this part by showing that we can do this on a biological sample. So we took the 1D spectrum in black from Neurospora crassa that I've shown you before, of the unfractionated extract. The red is the Batman fit of the metabolite basis set. We had 126 basis elements in this case. The blue is the residual, just about the same value of residual that we get for for the mixture, and you can just see different regions. The black is kind of bitmappy. This is not the resolution of the image. That Batman takes the thing and chops it apart when it's fitting this thing. So this is sort of a Batman artifact up here. It's the same spectrum that we have, but that's why it looks a little bit weird. But you can see that the fit's quite good. You can go through and find a few places where you have a peak that didn't show up or an extra peak that shouldn't be there. But for the most part, pretty darn good fit. The blue is the residual of the fit. And then here is the aliphatic region over here. So again, a pretty, I would say, good representation of the original data. And the key here is that we know every single concentration of all 126 basis elements from that fit. We might know 40 of what their identities are, but we know what the rest of the concentrations are. Okay, so that's what I have for the metabolite fraction library and using proton NMR. I'm going to switch gears in the last little bit and tell you about carbon-13 NMR and how that can be used to improve quantification in NMR. And this will go faster than the first part. So C13 is a really great nucleus. If you haven't embraced it like I have, you should consider it, especially if you have a 1.1 gigahertz available. So C13 detection at high field has a lot of advantages. First, I'll sort of lead with the disadvantage of proton. So proton frequencies at 1100 megahertz, proton interacts strongly, those frequencies interact strongly with the sample. So the ionic properties of the sample, the dielectric properties of the sample, both of those things degrade the signal to noise in a known way, and it scales quite steeply with frequency. So 1100 is much worse than 600, and it's still worse than 900 and 800. So it's getting worse and worse. Proton NMR at higher and higher frequencies is just becoming difficult. There are ways that people are trying to come up with to improve it. But in the meantime, carbon is the C13 is 1/4 of the frequency of proton. And so it's basically, less than 300 megahertz. It's a 280 or something. And so that's very tractable still. So 300 megahertz proton NMR is not influenced by salt and dielectric, and neither really is C13 as much. So it's much more amenable to high field NMR. And then we also have a probe at UGA that's optimized for C13 cryo probe called the TXO, and it can have a 5 millimeter sample, so you can get a lot of material in. So why would you want to do it? So here's some C13. So I've shown you this. These are the proton spectra of glucose, and I've highlighted the fact that these are overlapped. It's hard to analyze these things. So the proton chemical shift range is around 10 ppm, let's say. So let's see what C13 would look like. So here is a natural abundance C13 carbon spectrum at 1.1 gigahertz proton frequency of glucose. So you can see the peaks are sharp. They're not overlapped, and also not shown here because there's nothing in there for this spectrum. But carbon has a chemical shift range of around 200 parts per million, so roughly 20 times bigger than proton. And so there's a lot more room to put in really not so much overlap peaks. So these are great. Natural abundance samples are wonderful. They have single sharp peaks, very little overlap. But the problem is that only 1% of the sample is contributing to this. So what was already kind of lowish sensitivity now is becoming really low sensitivity. But that TXO helps. So it's actually very easy to get spectra at natural abundance for many samples. However, if you isotopically enrich your sample, you get much better results because you have more sample. You're not limited to that one. You have 100% of your sample is contributing to the signal. This is glucose uniformly enriched. But as you can all see, that comes with some costs, right? So you get enriched samples have greater sensitivity, but they have increased overlap because now we've introduced carbon-carbon couplings. These are all proton decoupled, but these are carbon-carbon couplings, which now kind of creates the problem that we were trying to fix in the first place with proton NMR, but we have a little bit more room in there. And so we have a really good probe to do this. But so we have to, so it's not perfect to isotopically label, but it's really good. So now I'll show you what we can do with some of this. This is in an adequate spectrum. Anybody other than Milo and Katie and Chad and Sam probably have never heard of inadequate spectrum. You have to be old and crotchety. No offense to you guys, but okay, yeah. But this is like the best spectrum, and this is the best NMR method in the world for structures of unknown organic molecules. It was invented in the 80s by Bax and Freeman, named by Freeman because he likes to come up with great acronyms. And I'm showing this spectrum not to brag, because you can hardly see the peaks in here. It was intentional, but to just show you this is what you get. This is natural abundance inadequate spectrum. It's one of the best ways to unknown, but it's very insensitive. So carbon is 1.1% natural abundance, and you have to have two of them together to get an inadequate spectrum. So this is 1.1 squared, which is about 1 over 8,000 of your molecules are actually contributing to the signal in this spectrum right here. So it's a pretty tough experiment. That's why nobody knows about it or does it. But you get a lot of really useful information. So here, for example, as you all know, glucose exists in two different anomeric forms. I've traced both of them. And you get the full structure of the back. You see which carbon is correlated to which carbon. Pretty tough to do. I mean, you can do COSEs and HSQCs and HMBCs and all this stuff, but this one single experiment gives you the entire backbone of a hydrocarbon, and it's very, very useful. So you can trace the entire thing, but it's low intensity. But then if you incorporate C13, this is the same uniformly enriched glucose sample. Look at that. You get tons of peaks in there with a much shorter acquisition time. And so and I'm using the same traces. But the interesting thing, those of you who know will see that some of the peaks that should show up in an inadequate spectrum actually don't show up very clearly in this one, but all sorts of additional peaks through other higher order carbon-carbon correlations do show up. So on the one hand, you can say, well, this is a mess. On the other hand, it's actually, I used to call these things bad name peaks. But now I'm realizing these are bonus peaks, because we can actually simulate these things directly using spinach density matrix simulation. We can simulate a spectrum that looks just like that from first principles. And so what it gives us, where we're going with this is we're going to start to simulate large libraries of these enriched mixtures, and not just try to search for these little peaks that show where the inadequate pairing are, but just say this is the signature of glucose. And we should be able to train machines to do this much better than we're doing it by hand. But we've been good at doing it by hand. We have a couple of publications out, and I'll just highlight very briefly that we can make a database. So you might say, well, there's no database for inadequate spectra, and there really isn't. But BMRB in particular has a bunch of high quality carbon 1D data that are assigned, probably half of them or more by Milo. And when you have an assigned carbon 1D spectrum, you can basically make an inadequate spectrum based on the rules of inadequate. So we have libraries of inadequate spectra that we then match to an experimental. In this case, our first publication was C13 labeled C. elegans. We've now done it on phytoplankton, and I'll show you a couple of examples of doing this on C13 labeled mice. So we've developed these different approaches doing that. So I'm just going to highlight in this last little part of this part before the E. coli, which is short, what we've done with a guy named Chris Turk from the Max Planck Institute. He came to me ages ago. It took a long time to get this thing going, but he had labeled 3 mice. These are just 3 normal mice. He labeled these things with C13 chow. That was the protein component was labeled. So not the carbohydrate or the lipid, just the protein component. These mice, he did it for mass spec studies, but what he found is that the C13 incorporation was so variable that he couldn't make heads or tails out of any mass spec work. He did this with N15, it worked great, but C13 was a lot more problematic. So he said, can you do anything with it? So I said, well, sure, we'll try. So he had these separate tissues and plasma collected. And so we basically prepared samples on 3 mice and collected data in our 900, or 1.1 wasn't there at the time. So 3 mice fed the tissues and harvested and extracted an inadequate, and then J-res, which essentially gives you a decoupled carbon spectrum. And this is what we got. So this is the C13 enriched samples. This is now the output of PyNeta, we call it, Python-based inadequate network analysis. And so these are all of the peaks in our original spectrum that behave according to inadequate rules, which I'm not going to go into, but they're very simple. And so we make networks. And so we had, I don't know, something like close to 100 different networks from the mouse liver. This is an inadequate spectrum from the liver. We have way more liver material than other tissues. And then the bottom is a JRES projection of the same sample. And we can take a network map and map it down to the peak in the JRES. And it's shown in more detail up here for lactate. So here's the inadequate network for lactate. And here are two of the peaks from lactate, C1 down to here and C2 over to here. C2 is a little bit off, but now these things are, we're just highlighting the C1 intensities of lactate across the different three samples. Remember, this is just to show that we could do something, but for different tissues and show the intensities. And then this is a table, for what it's worth, of the compounds that we were able to match to databases, all derived from BMRB. And then we have the nice thing here is that we have a bunch of unknown networks, but we know what the carbons are that are correlated together. So it's a good start for unknown compound identification. And then finally, these are compounds along the top that are enriched in liver. Relative to all the tissues. Again, this is N of 3, so there's no real biological thing except just where some of these compounds distribute where you would expect them based on metabolism. And then here are two other compounds that are enriched in other tissues that are not liver. And here are the type 3C that we're calling, which are enriched both in liver and a few other tissues. So kind of arbitrary categories. But we can do this kind of experiment with C13 NMR that would be difficult with other NMR methods. For the final, very short few minutes, I want to just show you what we were doing with C13. This is just a couple of slides. We had a user come to us and say, that they want to study, they want to know the difference, that they suspect that there's a different metabolism in E. coli grown in H2O versus D2O, and they want to know why. And the reason that they're doing this is that they have a technique that they use, put the organism into D2O, and they can determine antibiotic resistance from that. And so the problem is that proton NMR doesn't work if we want to compare D2O to H2O. LCMS doesn't work because everything's going to change mass. So how do you do it? Well, obviously, C13. So Leandro grew up a ton of samples, basically M9 minimal media, uniformly labeled. C13 glucose was the sole carbon source, 15 time points collected every 2 hours, measured the OD and the pH, separated the media from the cells and measured it. So that's what we got. And then this is it's easier to see this just in a video. I love this video. So this is showing on the bottom is the media, and on the top is the pellet, the endometabolome, the pellet. And so you can, and then the little ball is going along both the OD change and the pH change of the samples. So the pH is on the top is dropping now rapidly. We're reaching full optical density. So you can just see the evolution. This is just one matched pair out of our 5 replicas of media and pellet. And you can just see, so the glucose is disappearing. Here's glucose, it's being consumed, it's being turned into other metabolites. So that you can get this kind of level of detail with direct C13 NMR. So what are they? So Leandro has been able to match roughly 50% of the peaks that we get in there, either direct match to BMRB database with 1D or with inadequate. A few of the time points we collected inadequate. And this is what they look like. So this is the ones, so about 50% more that are not on here right now, but you can get this kind of detail. So we're showing now the concentration of the peak or the amplitude of the peak across time, and they're roughly ordered in the intensity of the peaks. So tons of detail on the flux that are in there, and all the time course detail, and then we have a corresponding media time series. GABA and glucose dominate the media. So this first slide shows those things. This now shows without those two, and a similar sort of time course of other metabolites. We actually know a lot less about the media right now because it has a little bit more pH variability and some compounds that we weren't necessarily expecting. So we're trying to take this data, put it into E. coli type pathways and try to use it for flux balance analysis type applications. Okay, that's it. I just want to summarize that overlap in 1D NMR really is a huge problem. I think a bigger problem than many people who do NMR routinely realize. Metabolite fraction libraries, I think, is a really nice way to reduce the overlap. Bonus from that is that you get metabolite basis sets, and then that replaces the need for a database to match your profiling data. And it's just the right size because it's what you see. C13 is really advantageous in high fields, and things like inadequate can help quite a lot, especially with machine learning. So the funding agencies, we're all grateful for all of the federal funding that we can get these days, and we especially really thank NSF. Was a heavy lift for Nan in this, and we appreciate that. Chad and Katie and Jeff Hoke at UConn, and then the team that I have at UGA, Chris and Frank and Gonzalo and Bree for fraction libraries. These are the folks that were instrumental with this Pinetta approach, the inadequate, and Leandro mostly for E. coli flux. And then the final acknowledgement, I especially want to give thanks to John Markley, without whom I would never be here right now, and Frank Weinhold, who were co-advisors during my graduate career. I mostly calculated NMR coupling constants with Milo, but Milo taught me everything about NMR. So if I had any mistakes, it was because of Milo. But we spent years doing product operators on his chalkboard and setting up complicated experiments before they were ready to do. And then Eldon, as all of you know, really started the BMRB. The BMRB became what it is because of Eldon, who passed away prematurely. We think recently. Memorial services is Saturday, right? I'm sad that I'm not going to be here. And I'm really grateful to Professor Everson for endowing this seminar series, and for Chad and everybody for inviting me. So thank you.
Okay, are there any questions? Ben, looks like.
Thanks, Art. That was a great talk. In your metabolite, the first part of your talk, you showed the residual between the experiment and the fit spectrum. Can you use that residual to help better fit, or use that to help you, improve the accuracy of your simulations?
Yeah, well, so let me just correct the term simulation a little bit, just less. These are simulations, but they're basically Bayesian fits of the metabolite basis set. So
that's with the Batman, right?
Yeah, the Batman is it's using a statistical. So basically, the prior knowledge are those metabolite basis functions. So yes, we can use the residual. In fact, Chris used the residual extensively to try to come up with. You have to adjust. There are a lot of adjustable parameters in Batman, and he found conditions. The biggest thing you have to do is Batman will allow for chemical shifts to move, so it will account for pH changes in the sample, and that's really the thing that it was the biggest thing that took some time to dial in, and we didn't quite get it for the histidine sample that I showed.
So you can take Batman and optimize it to minimize your residual?
Yeah, Batman is a complicated program that has a lot of residuals, but we shared these results with Tim Ebels, who made Batman, and asked him for his input, and he said, wow, that's the nicest fit I've ever seen using Batman. So I took that as a good sign, but he did help us a lot in getting it started. I thought you were going to ask, we have a study where we're comparing mutants and wild type, and we have a fraction library for the wild type, but not for the mutants. But we're actually looking forward to using the residuals in that case to try to ID or find peaks that are unique to just one or the other, but not both. That's where I thought you were going to go, but that's what we're doing too.
Cool.
Yeah.
Thank you.
Yeah, thanks for a really interesting talk. So when you were looking at the inadequate spectra, and you had, I guess, an inadequate spectra of an unknown compound, and you were using the 1Ds to, I guess, simulate the 2D inadequate, how are you comparing the two?
So you mean the software that we've used to do it? Basically, so the rules of inadequate are if you have two carbons that are bound together, let's call this one 10 ppm, this is 50 ppm, they show up at 60 ppm, the sum of the two frequencies. So if you have assigned carbon spectra, you have exactly the inadequate spectra. So there's no approximation with that step. There's known quantum mechanical rules that will give us the inadequate spectrum if Milo did his job right and assigned the carbon spectra right. Not to always pick on Milo, but so then you can just overlay that on your experimental spectrum. The thing that is difficult is that, like you saw in the glucose enriched sample, some of the bigger peaks are not following this rule. So we actually don't see them in those inadequate, but we do see peaks usually in the rule where we would expect based on the rules of inadequate. And we can pull those out of mixtures. And this software, if you're interested in this Pinetta is on GitHub, and you can just download it and try it out. We can collect an inadequate spectrum for a sample if you want, and you can try analyzing if you have a mixture that's enriched.
All right, thanks.
Yes, it was great. Sit down.
For the E. coli flux, when you were looking at the change over time in the growth, how much of the change in metabolism or in the media components that you're seeing do you think is because of the change in pH versus actual growth and changes, say, from exponential to stationary phase?
Well, so the change in pH is known.
Right.
So that's something that happens, and every person that I've talked to said, yeah, that's exactly the way it should be. So I guess I don't know.
If you were to repeat the experiment with buffered media so that the
Oh,
pH isn't changing,
It's buffer. I mean, it's M9.
well,
It's
there's buffered, then there's.
okay. I mean, I don't know if we would get exactly the same thing in like an LB
Mhm.
media or something. We haven't done that because mostly just because we wanted to get carbon labeling. I'm not positive about that, but they do grow. I mean, they grow more slowly in M9 minimal media, but at least we've always attributed that to the fact it's M9 minimal media and limited just glucose.
Mhm.
So that's, but they still grow happily. So I don't know. Yeah, are there going to be metabolism changes between M9 minimal media and LB? Yeah, I'm sure. And that's for another day, I guess, for us.
Art, I had a question about histidine because it sort of seemed ironic that you used it for the quantitation standard at the beginning.
Well, we knew where
And
it
then
was
when
at
you
that
fit the
time.
mixture, it's like the worst one, right? So
Well,
why
so-
is that?
So, okay, so this actually is sort of, it's not related to Katie's, but it's the same phenomenon. The mixtures, we ended up in our wisdom, my wisdom, because I said, nah, don't use a buffer. We don't need it. These are synthetic mixtures. And it turned out that we should have used a buffer. So they were unbuffered. They were unbuffered mixture spectra, or at least 2 weekly buffered. If there was, I think there might have been a little bit of phosphate buffer, but it didn't do a very good job. One of the spectra had histidine that had shifted way out, and it was just too hard to sort of tame it and pull it in. Yeah,
So it always comes down to the sample preparation.
yeah. It's
And
always
usually
sample
then the
prep,
buffer.
even for
Yeah.
standards.
But even with the carbon spectra, it wouldn't resolve that, right? Because the carbon would be
Well,
less sensitive to pH.
Yeah,
or easier to
carbon,
resolve.
one of the huge advantages of carbon is it doesn't have nearly this pH swings that it does, but we didn't collect carbon 1Ds on these things either. So we probably should have, but we didn't.
All right. All right. Well, let's thank Art again for a nice talk and
Thank you.